Hi, dear students. Welcome to the new session of microbiology. And today we are going to talk about the early microscopy. One of the major constraints of microbiology is that microbes are quite often of small size and they are not visible to our naked eye. So, we need certain instruments such as the microscopes to view them. And the advancements of microscopy is a major breakthrough in the field of microbiology. And all the advancements which we have made today is mainly due to the advancements of microscopes. So in this session, we would be talking about the early microscopy and the efforts which were being made in this direction. The discovery of microscopes was mainly being done in 1590s when two Dutch spectacle makers, the father and son team, Hans and Zacharias Janssen, they created the first microscope. These microscopes were compound microscopes which use at least two lenses, and the objective lens was positioned close to the object and it produces an image that is picked up and magnified by a second lens called the eyepiece. So, a compound microscope is a microscope which uses two lens systems. And the first microscope, which was being discovered by Zacharias Janssen, was mainly a compound microscope. So, the first objective lens based image was being further magnified by the eyepiece lens. And you can see a reproduction of the first compound microscope, which was being made by Hans and Zacharias Janssen. Uh, in 1590, and this is a picture which has been provided by the National Museum of Health and Medicine, Washington, D.C. And later on, the microscope which has been developed by them was being further improved by Galileo in 1609, and he called it this particular device as occhialino, or the little eye. After that came another scientist called Robert Hooke. Robert Hooke was an English scientist and he also improved the microscope, and he started exploring the structures such as snowflakes, fleas, lice, as well as plants. And he was the one who put forward the term cell from the Latin word cella, which means small room. He compared the cells which he saw in the cock to small rooms that monks lived in. So when he took the cocks of plant cells, he found like certain cells were there, cella, or certain compartments or small rooms were there. And he computed them and he named them as cella, cella based. That means the term cell was being given for the small rooms which he saw in the cock cells. And in 1665, he detailed all his observations in a book called the Micrographia. So the discovery of cells was mainly been done by Robert Hooke, and he used a compound microscope, which was further modified from the previous versions. But the compound microscope, which was used that time, also had severe chromatic distortions because what happens compared to the single lens systems, the compound microscopes caused aberrations or the distorted images were being formed when we used early compound microscopes. The Robert Hooke, he was the first one to report the cell structure in 1665, and it was the first illustrated book on microscopy. Other than the cock cells of the plants which he had observed, Robert Hooke also observed microorganism, mainly the fungus mucor, in his studies. So all his observations were being encoded and enlisted in the book in Micrographia. Now comes another eminent scientist, Anton von Leeuwenhoek. He was the first person to see bacteria and explain it accurately in 1676. Unlike Robert Hooke, Liebenhaus was mainly using a simple lens microscope, and it was made up of two convex lenses which have been held together in a between two silver plates. And the Liebenhock made microscope consisting of a single high quality of lens of very short focal length, and at a time such simple microscopes were being more preferable than the compound microscope because the chance of chromosomal aberration or the distortion of the images were being found to be less in the case of Liebenhock's microscopes. But who is he or what was he? Basically, he was a Dutch businessman and a scientist in the golden age of Dutch science and technology. And because of his efforts, which he has made in the field of microscopy as well as in the field of microbiology, he is being termed as a father of microbiology. And he is known for his pioneering work in microscopy and for the contributions towards the establishment of microbiology. Now, he did not write any books, and what he did was all his discoveries were being, in fact, sent as correspondence with the Royal Society, mainly as letters. And all these letters were being further published by the Royal Society. Though he didn't write a book, but it was his letters explaining his discoveries that laid a foundation for the development of microbiology. And what are his observations? In 1674, he likely observed the protozoa for the first time, and many years later, he also observed bacteria. At that particular time, he called these as very little animalcules, and the term animalcules was the one what he used for it. And he isolated them from the rainwater, pond water, as well as the well water, and sometimes even from the bacteria which were being found in between the teeth. That is, after drinking a cup of coffee or something, he would take a scraping from his teeth and he used to observe it. As well as from the intestine also, the bacteria was observed. And not only observing, he also calculated the size of those bacteria. And he made first time spermatozoa observation of spermatozoa was being done from insects, dogs, as well as from man. And his microscopes produces images which were about 270 times larger than the actual size. And he was the first to document the microscopic observations of muscle fibers, the red blood cells, as well as the blood flow in capillaries. Now, this is a picture depicting his microscope. You can see it's a single simple lens system. Okay, it's just on either side. How does it look? This is one side of the picture and the other one is the other side. So here he has a sample holder. It's like a point, it's a it's a pointed like thing, and on top of that he will place the drop of water, or if he's a drop of water, well water or pond water, respectively. And this is the focusing knob which is being used. Now these are the images which he has obtained the spermatozoa from man, dog, and all that. And as I mentioned earlier, he also studied yeast cells, red blood cells, as well as bacteria from the mouth and the protozoa. So with this, we come to an end of the early microscopy. The efforts, the three main scientists who played a main role in the field of microbiology. Of course, the discovery of microscopes was being done by Zacharias Janssen, and further than we had Robert Hooke, who observed the cells and coined the term cell. And then we come across the father of microbiology, the great Anton von Leeuwenhoek. So with this we come to an end of the session. We will be continuing in the next classes. Thank you for now.